Thank you very much, Daniela, for this kind introduction, and thanks everyone for joining. So, as Daniela said, I'm a group leader at Center for Neurogenomics. I'm originally from Croatia, where I studied molecular biology at the University of Zagreb. But I got very early interested in molecular and cell biology of neurons. And this is actually what led me to do my PhD at EMBL, where I studied the acetylation of microtubules in somatosensory neurons and how this affects their function. And already during my PhD, I was fascinated by the development of the nervous system. And that led me to do my postdoc in the lab of Wieland Huttner at Max Planck Institute of Molecular Cell Biology and Genetics in Dresden, where I was focused on understanding the development of the human neocortex, which is one of the key structures underlying our cognitive abilities. And I was also interested in understanding the evolutionary connotations of this structure, since neocortex is indeed the newest part of our brain, and it underwent a massive expansion in human brain evolution. I was very fascinated in understanding the molecular and cell biological underpinnings of this expansion. And these are also the main topics that I'm carrying on with me that I will now study in my own lab at Human Technopole, which is going to be focused broadly on molecular and cell biological mechanisms that are underlying human neocortex development and all the implications this can have on evolution and human pathologies, where I'm particularly focused on neurodevelopmental malformations. As you can see, this is already very multidisciplinary question, so also our techniques and approaches are such. And we combine a variety of different model systems, precisely in line of what Giuseppe was saying before, to find the appropriate model for specific aspects of humanness. And I use various in vivo models, such as mouse and ferret, whose brain cross-section is here on this slide. And you can immediately see the power of ferret, because it can clearly replicate one of the key characteristics of human brain, which is this characteristic folding pattern, which is something that a mouse model, for example, cannot. In addition, we combine this with various human model systems ex vivo and in vitro, and molecular and genetic techniques that are notably based now on CRISPR-Cas9 to functionally perturb this. One of the key biological findings from my postdoc was that morphology of neural stem and progenitor cells seems to be one of the key parameters underlying their proliferative capacity, that is their ability to make neurons, and thereby drives the expansion of neocortex through development and evolution. And this is something that I'm now building on, also to look in context of human neurodevelopmental pathologies. And here we are really interested in two types of disorders: those that are caused by defects in proliferation, that is the ability of neural progenitors to make increased or reduced number of neurons, which then leads to increase or reduced size of the brain and causes disorders like microcephaly and macrocephaly. And on the other hand, we are interested in those malformations that are caused by defects in migration, where the mutation can be present either in neurons or in the actual progenitors, whose morphology serves as a scaffold for neuronal migration, and can be cause of disorders such as lissencephaly, polymicrogyra, and heterotopias that will all affect the shape of the brain and often the folding of it. And as you could see, this is quite interdisciplinary questions, and they are, as such, it's very important for me to be embedded in this center of neurogenomics headed by Giuseppe Testa, and to really address the human experimental neurobiology in context of malformations, in my case, at proper scale, at which we are aiming to do, combining these various models and techniques that we try to quickly show here. But of course, the questions are also even broader, and because of that, it's very important to benefit from interactions within the institute, and we going deeper into the mechanisms governing genome regulation is something where, with the Center of Genomics, it's very interesting to move forward, and especially in context of structural biology, which is now following in this presentation. I'm very interested in going into the molecular organization and structure of the complexes that are regulating morphology in neural progenitor cells. And finally, many aspects of data and image analysis is something we are closely collaborating with the computational biology. Finally, I'm also very enthusiastic to be moving to Italy, and about the prospect of establishing synergies with the Italian life science community. And I'm also hoping that this event and similar events are going to be helpful in bridging this and establishing good networking. And with that, I would like to thank you for your attention, and I hope to meet a lot of you in person soon.